everyone, and thanks for tuning in to BEaTS Research Radio. I'm Hallie Arnott, your host on today's episode, and today we have a very special guest with us. We have Dr. Mohsen Akbari, who comes to us from the University of Victoria in British Columbia. Dr. Akbari completed his PhD at Simon Fraser University, followed by a postdoctoral fellowship at Harvard Medical School, and is currently an associate professor at the University of Victoria in the Department of Mechanical Engineering. He is also the principal investigator of the Laboratory for Innovations in Microengineering, or LIME for short. Dr. Akbari's laboratory lies at the interface of cellular biology, biomaterials, and mechanical engineering, where he and his team are particularly interested in combining the fundamentals of fluid, mass, and thermal transport on the micro scale with cellular biology and biomaterials to address current challenges in global health. Three key research areas in his lab include tissue engineering and regenerative medicine, advanced drug delivery systems, and biomimetic tumor models for drug discovery. Thank you very much, Dr. Akbari, for joining us here today on BEaTS Research Radio.
Well, thank you very much for inviting me to this very interesting podcast and interview. I was watching some of the videos, and then I learned a lot from the conversations that you folks had with the researchers, and it's a pleasure to be talking to you folks. And thanks for the kind introduction.
Yes, absolutely. I love learning lots of different things every time I interview somebody new. So we'll get going with today's interview so I can learn a bit more about what you and your lab are doing over in BC. So before we get going into the research aspect, I'd like to ask you, I read that you began your career in the automotive industry before transitioning into research in biomedical engineering. What made you redirect your career pathway towards this field?
Well, that's a very, very good question that I really want to talk about a lot. When I did my bachelor and master's in mechanical engineering, the main focus of my research was in heat transfer and fluid mechanics, with the focus on designing heat exchangers for cars. So I was very much involved in a theoretical analysis of heat exchangers and condensers. And then I published a few papers. But then when I moved to Canada to start my PhD, I was introduced to the field of microfluidics. So I had two supervisors. One was in the field of energy, and then the other one was in the field of, he was working in both fields of energy and health related basically research. But his focus and expertise was on microfluidics. So when I was exposed to both of my supervisors, I learned, of course, from each of them. But I always had the passion to do something in the area of health. The reason for that is because right before I moved to Canada, I lost my mother to cancer, and then she was fighting with breast cancer for almost 6 years. And then when I was with her, I was always thinking about what could I do to for patients like her, to first find a way to treat them better, or at least make their quality of life much better. That was what I was thinking about during the 6 years before she passed away. And then with that in mind, and after I was introduced to microfluidics, and then also some of the health related research that was going on in both labs, then I decided to slowly shift towards more health-related research, although I was still focused on fundamentals of heat and mass transfer in macro scale. But I was also had this on the back of my mind. But then I had this opportunity of that I received an NSERC fellowship, which gave me 2 years of support to continue my research. And so that gave me a little bit of flexibility to choose where I want to do my postdoc. And then I decided to go to one of the like top labs in the world, which was Dr. Khadim Hosseini's lab at Harvard. And then I moved to his lab, and then I completely shifted gears, and then I shifted my research area, and then I became familiar with the concept of tissue engineering and the use of microfluidics in tissue engineering and drug delivery, and also some of the concepts in 3D bioprinting, organs on a chip. I was exposed to all of these great research projects that were going on in such a huge lab. It was a mega lab that I was that I joined to, and then I learned plenty from all these high caliber postdocs, and of course my mentor Ali K. We called him Ali K. And then that's how I completely switched my research direction to biomedical engineering. I mean, one thing that I need to mention is that before I joined Harvard, I spent a year at McGill University in David Younker's lab. He is an inspiring mentor, and then that transition from from SFU to Harvard was actually like that intermediate step that I spent some time at McGill was very useful for me, because I was exposed to like one of the smartest people I've ever seen in the world, and then I learned from him how to think about science, because I was an engineer, but he was a scientist. So engineers, I usually say that engineers, they try to solve problems, but they don't, go that much into details. But scientists are more detail oriented, right? They go deep into the concepts, and so combining my engineering expertise with this detail oriented, approach that David was following was very helpful for me, and it made me ready to go and then jump into a big lab, which was an excellent experience, and that's how that was the journey.
That was the
And I
journey.
decided to continue, my research in the area of biomedical engineering.
Mm-hmm. Yeah, it's incredible to hear those kind of stories and have a career completely shift and be successful in both areas equally the same, and that's great. And also to hear all of the collaborations and how you can have a totally different expertise than somebody else and still be working in the same field. And I think that's how biomedical engineering works well, is when you have somebody like yourself who's come into the field but with an incredible skill set that's very valuable, that can be combined with other scientists, as you said, and that's great. So now to get into some of the work that you do, one of the areas of expertise when I was reading about your lab is the development of functional 3D tissue constructs resembling hydrogel fibers. Could you explain to us how you do this, and what is the end goal for these 3D tissue constructs?
One areas of expertise in my lab is to use microtechnologies to create functional tissues. And we are in particular interested in fiber-based tissues and making fibrous structures. And the reason for that is because fibers there are two reasons. First of all, there are a few tissues in the body that have fibrous structures. So in the field of tissue engineering, what we do is we look at the tissue, we look at the organ, and then we see how we look at the structure of the tissue in terms of the microstructure and then the cytostructure, the cellular structure that exists in that organ. And then we try to mimic it using the engineering tools that are available to us, engineering and of course chemistry tools that are available to us. And one of the tissues of interest for us is skeletal muscle, and then also neural tissues which have fiber-like structures. So they are formed from bundles of fibers that are organized that are put together in a very organized manner, and then they work in a way that's in a highly synchronized way, so that, for example, in the case of muscle, you can contract your muscle and then perform or apply forces when they're needed. So we look at these types of tissues, and then we see that all of these tissues are formed from modules that are fiber-like structures. So they are very long, one-dimensional fibers that are bundled together to form all these tissues. And then we thought about how we can use the technologies that are available to us to make all of these fibers. So we thought that maybe we can make all of these small fibers using microfluidics technology, and then this microfluidic technology is something that was not actually invented in my lab. It has been out there for a while now, for almost a decade, but the idea is to generate fibers out of hydrogel materials using coaxial flow systems, in which you generate fibers in a microfluidic device to form two streams or multiple streams of materials in a microfluidic device. And one of the streams is the prepolymer, which can be encapsulated with cells, and then the other stream is the cross-linking agent that helps these fibers to cure and then to polymerize, and then eventually you can collect the fibers at the end of this microfluidic device. So this is the general idea that we use to create functional fibers. However, we work on we work on creating fibers with various shapes and various morphologies. For example, recently we were able to create functional fibers made out of hydrogels with microstructures, structured grooves on the outer surface, or we were able to create hollow fibers with internal grooves, and also we were able to create multifunctional fibers made out of multi-materials. So this microfluidic technology enables us to have a very good control on the material composition and also structure of the fibers that we can generate for different applications. So these fibers are not only used for tissue engineering applications, we are also using them for biosensing applications, and then also for drug delivery applications. For example, we have made fibers that are electrically conductive, and then when you apply a voltage, these fibers can heat up, and then if you now load them with temperature responsive polymers, which are loaded also with some drugs, then by applying an electric field, then you can heat them up, and then you can release the drug content on demand when you apply this electric field. So this is the kind of work that this is the way we make fibers. We make these multifunctional fibers, and then now this microfluidic device, you can either collect the fiber on a spool, and then you can imagine that you can have multiple spools of different fibers, which could be drug-releasing fibers, cell-loaded fibers, or electrically conductive fibers, and then use available textile methods such as braiding, weaving, embroidering to create one-dimensional, two-dimensional, and three-dimensional structures. Or you can just simply flip this microfluidic device and on an XYZ control stage, and then you can print now deposit these fibers on the stage in a way that you can achieve a 3D printed structure with controlled properties in terms of the morphology and of course the cellular distribution. So this is the kind of work that we do in my lab. And
Wow.
yeah.
That's very interesting. So these microfluidic devices you can keep. So you were saying you can either keep the fibers in the microfluidic device, and then is it possible for, sometimes you take them out, but is it possible to create a chip where the fibers are created in there, and then you're also able to achieve a three-dimensional sort of construct in the microchip, so that you can maybe use it for different, like in hospitals or something, and I guess do any sort of like organ on a chip work?
So that's a very good question. Yes. So one of the applications of 3D bioprinting is to combine it with microfluidic systems. As I mentioned, there are two ways. So there's one way, one product I would call them is that comes out of these chips is just a single fiber. It's like the threads that you have, yarns that we use in our cloth, right? So imagine that these yarns with different colors you see here, each of them could be have different materials, different cells, different properties, right? So we can make these fibers, and then we can either use these fibers individually in a microfluidic device to, for example, mimic the blood vessels in the case of making hollow fibers. So let's say we make hollow fibers, and then we coat the inner surface with endothelial cells, so we can have a blood vessel, which is functional. And then you can implant this blood vessel in the body, or you can put it on organs on a chip system to mimic the vascular system, the blood vessels, and so on. The other way of using this technology, combining this technology with organs on a chip, is to make these chips, these microfluidic devices, and then and again use this microfluidic device as the stage, as the XYZ control stage, and then print or deposit fibers in a perfusible chamber. So with that, you can create three-dimensional, 3D bioprinted construct in a microfluidic chamber, and then you can use this microfluidic chamber to continuously perfuse the bioprinted construct. So the benefit of perfusion is, of course, it's more biomimetic, because in the body we have organs that are continuously being exposed to nutrients and oxygen, to the blood flow, and this will help us to maintain the fabricated tissues for longer times, for long-term studies. For example, if you want to investigate the long-term effect of different drugs on a certain tissue, then you can use this artificial or this organs on a chip platform to study the effect of these drugs in the long term.
That's incredible. So it's an incredibly translational research group that you have going. Is there a project that maybe is ongoing that you could tell us a little bit about, where you're using this organ on a chip model, or the microfluidic and also the bioprinting combined? Is there something your team is currently working on?
Yes, we are right now working on two tissues. One is skin tissue. My lab is heavily focused on developing advanced drug delivery systems and advanced wound dressings for management of chronic wounds. And then part of the research part of this research program is developing full thickness skin, fully functional skin models by which we can test these drugs or these wound dressings in vitro. So the goal is to create a more biomimetic tissue, skin tissue, for safety and efficiency studies, so we can reduce the number of animals that can be used for preclinical studies. So that's one of the tissues that we are working on. And then we use bioprinting to create the skin tissue. In particular, we use bioprinting to create vascularized tissue, because vascular network is one of the most important components of of the skin, and it's actually very it plays a key role in the wound healing process when the macrophages and the immune cells are being recruited through the blood vessels, so they will they will cross the walls of the vessels, and then they move towards the injury to start cleaning up the bad stuff at the wound, and then start the healing process. Another research area in my lab is focused on brain cancer. We are specifically focused on glioblastoma, which is the grade 4 glioma cancer. This disease does not have any treatment. There's only it's pretty fatal, actually. So the life expectancy for patients is almost 1 year. So only 5% of patients survive after 5 years. So it's a pretty deadly disease. And there's only one drug available at the moment for treating patients, and that's called temozolomide. So it's just a pill that the patients need to take, but it's pretty, it's not very effective. And the reason for that is because brain has something that is called the blood-brain barrier that does not allow most of the drugs to penetrate into the brain tissue. So basically, we have this blood-brain barrier that prevents the drugs to reach the cancer cells and kill them. The other issue with the brain cancer is that the brain cells, it's unlike some other tumors, brain cells are pretty invasive. So the brain tumor, you see a tumor mass, but there are finger-like basically branches that branch out from the actual mass, and then they go deep into the brain tissue, and then sometimes the depth of penetration is 2 or 3 centimeters. So removing all these cells that are deep, 2 centimeters deep into the brain is almost impossible. And so there must be new drug delivery systems that can deliver the drugs deep into the brain tissue to reach those cells that went, that are 2 centimeters away from the border or from the tumor mass. So to understand how this drug delivery system, how this drug delivery system should work, and to understand the mechanism of tumor cell invasion and progression, we have developed a microfluidic device combined with bioprinting that mimics the tumor mass and then the stromal layer. So we have this, we create tumor little organoids or cancer organoids from cancer cells, and then we embed them in a matrix that mimics the matrix of the brain. And then the matrix has, of course, all these healthy cells. So the matrix is modeling the brain, the healthy brain tissue, and then this tumor or cancer organoid mimics the tumor mass. And then with this tool, and then of course with the microfluidic device, we can model the invasion of tumor cells into the healthy tissue, and then we can quantify how fast these cells are invading, and then how far they are penetrating into the healthy tissue. And then we can use this as a tool to investigate the effect of different drugs or different treatment modalities, because chemotherapy is not the only modality that is being used for treating glioblastoma. Chemotherapy combined with radiotherapy is now the current clinical practice for management of glioblastoma. And then, of course, there are some other new drugs that may be able to cross the blood-brain barrier, so we can test them. So this bioprinting combined with microfluidics provide us with a powerful tool to use to screen different drugs and then investigate their safety, and then also their efficacy, and then of course their safety, because you can use whatever drugs that you want, but you have to make sure that you don't kill all the healthy cells too. And then that's another thing that we need to we need to make sure that that these drugs are not harmful to the surrounding tissue, especially for the brain, which is a very sensitive organ.
Right. Yeah, definitely.
Yeah.
Oh, yeah. So you're basically making the platforms that are going to enable better production of these drugs that are going to help these patients. So that's very, very amazing. So there's this other project I wanted to ask you about. It was a smart bandage that I wanted to bring it up because I thought it's very interesting and very cool. So your collaborators at Harvard Medical School and your lab worked on creating a bandage that was able to detect and treat infection using a smartphone app. Could you tell us briefly about that project and what happened with the study?
Yes. So the idea here is that I mean, wound management is a pretty old field, and then if you look into the literature, you see that there are thousands of different uh, wound dressings available in the market. But the focus of pretty much all of these wound dressings is there are two things. Their goal is to either protect the wound from infections. The other one is to improve the healing process. So the way they protect the wound, so for the first goal, what they do is that they provide a barrier against the external pathogens. And if the wound is infected, then some of these dressings, they have drug delivery capabilities. So they have antibacterial, basically, features that kills all the bacteria that may grow in the wound. And then there are some dressings that they deliver growth factors and then factors that actually improve the healing process. However, there is a big challenge in wound management, and that is called infection. So chronic wounds and infections are major. Infections in chronic wounds is a major clinical challenge. You can, of course, use antibacterial agents, but that will cause antimicrobial or antibacterial drug resistance or bacterial drug resistance. So the resistance is a huge challenge. Antibiotic resistance is a huge challenge right now in the field of wound healing and wound management. And infection by itself is also a big challenge. So infection can cause many issues. It delays the healing process. So it can keep the wound at the inflammation stage, which means that the wound never heals until the infection is cleared from the wound. So the delay some wounds, especially the chronic wounds, can heal as long as 2 or 3 years. So that could cause a lot of pain, and then also financial burden actually on the family, and then also on the healthcare system. The other issue with infection is that infection eventually can, if in serious cases, it can cause sepsis, which can result in death, or in some cases, it can lead to amputation. So these are the kind of issues that infection cause in the wound in chronic wounds. So what wound professionals do is that they usually ask the patients to come go and visit them regularly. So what they do is they remove the dressing, they look at the wound, they look for signs of infection, they even smell the wound to see if, they can smell, the wound smells different than a normal tissue. And then if they are suspected, they collect swab samples and then send it to lab to determine whether the wound is infected, and then also determine what type of infection does the patient have at the site of injury. So this is a long process. Imagine that most of the patients with chronic wounds are elderly. so they have mobility issues. We imagine that patients who live in remote areas, indigenous communities, they may face this issue of chronic wounds, and then they have limited access to centralized hospitals. So because they, of course, living in remote areas, and then imagine that in the situation that we are right now, in COVID, in pandemics like this, going and visiting a doctor is not always the safest thing to do, and then there are so many limitations for patients to go and visit their doctor. So so with that, going and visiting doctors regularly is not something that is easy for these days for many of us. So what we thought about was that we decided to come up with a smart dressing that has embedded sensors in it. So it can tell you whether the wound is infected or not. So these sensors are color changing sensors. So they don't have any color. So they're like transparent, no color. So you see that the dressing has its own color. It could be transparent or any other color. And then if there is any sign of infection, if the wound is infected by any type of bacteria, then these little sensors start changing color. So you see that in our case, they turn from transparent to a new color, which is completely visible by the naked eye. So if a patient sees such thing in the dressing, so they can take a picture from the dressing, they can send it to their doctor, and then they can set up an appointment with the doctor, so the doctor can ask them to give them ask for more information from them. So the doctor may ask them to remove the dressing, to take a picture of the wound, and then send them more information about the wound. And then this dressing is now at the moment it has sensing capabilities, but we are also working on dressing that have drug releasing capabilities. So the doctor who sits in his or her office can press a button and then release a drug at the site of the injury. So that's the ultimate goal of our smart dressing, which we have made a tremendous progress so far, and there are a few papers coming out pretty soon on this autonomous, we call them smart dressings, that they have a closed loop system that not only senses and measures monitors the wound condition, but also delivers treatment to the site of injury.
It's incredibly interesting. I think that kind of project brings together a lot of your expertise in fluid mechanics and also biomedical engineering. Also, all the projects we talked about today I learned a lot. So thank you very much. I think that concludes our interview. I thank you for joining us on BEaTS Research Radio. We are very happy to have you on our show today.
Thank you very much for inviting me, and I really enjoyed our conversation today.
Great. And thank you to all of our listeners. BEaTS Research Radio is supported by the University of Ottawa Heart Institute, the BEaTS Research Lab, and the Department of Biochemistry, Microbiology, and Immunology in the Faculty of Medicine. Our director is Dr. Emilio Alarcon. Thank you for tuning in, and don't forget to subscribe to our YouTube and podcast channels. We'll see you all soon with some more science in our upcoming episode.